Hi, Blazers. Dr. Raz again. I'm going to talk a little bit today about meiosis. This is section 3.3 on your PowerPoints, and I will load not only the PowerPoint, but the Screencastify this version onto our Google Classroom for us. So anyway, with meiosis, remember, we have two types of cell division: mitosis, which looks at the division of the autosomal or body cells, and meiosis, which creates for us what we consider to be haploid cells. You see this one N down here, this one N numbering system that produces four of these haploid cells. Now, with the egg, only one really is viable of the four, but with the sperm, all four would be viable. So we're looking at a diploid cell, which is what we're going to start meiosis with. We're looking at 2 times n being for humans 23, so we end up with our 46 chromosomes. This, of course, we're looking at after fertilization. Once the egg brings their 23 chromosomes and the sperm brings their 23 chromosomes. So we're going to take a look at all of these, the different phases of meiosis, so that we have a very good understanding of what exactly is going on. Now, remember, meiosis, is really important as far as sexual reproduction goes. We're going to run through our stages or phases twice when we're looking at meiosis, so that we can end up with cells that are haploid in number. We'll have a half the number. So if we're looking at the production of the sperm, which is, of course, spermatogenesis, we are looking at the end of meiosis 2 of ending up with our 4 haploid cells, all 4 sperm being viable. But if we're looking at egg production, which is oogenesis, then we're looking at the end of meiosis 2, ending up with 4 haploid cells. Only one of them, however, is going to be viable. The other 3 are not. So a lot has to go on, right, for fertilization to occur. So take a look at how we get to the point of fertilization. So even before meiosis can even begin, remember, we have to undergo, the cell is going to undergo interphase, which is kind of an introductory phase to the cell division, which in this case happens to be meiosis. So during that phase, there are 3 parts where the cell grows and where more DNA is made. So it's that S phase, which is the synthesis phase, where more DNA is going to be made. And what happens is we end up producing 2 genetically identical, what we call sister chromatids. We can see here's sister 1, here's sister 2, and then they become joined at the midsection, and we call that midsection, of course, the centromere. So our chromosome is made up of 2 sister chromatids that are joined together. And then we have the homologs, right? 2 chromosomes that are similar in size, structure, gene location, and so on. So all that has to happen before we even begin meiosis 1. Remember, meiosis is going to be split into 2 phases, right? Meiosis 1 and then meiosis 2. So all of this is done ahead of time before meiosis can even begin, right? We looked at that supercoiling of the DNA around the histones. So all that has to come about. So let's take a look at our stages of meiosis here, right? We always begin with that kind of introductory phase, which is not part of meiosis, but where the cell begins to grow, the sister chromatids connect and form chromosomes. So we're able to see the actual chromosome, of course, under a microscope, that appears. So the nucleus begins to break down, and we will see individual chromosomes inside. So notice there is meiosis 1 and meiosis 2. We have to have two phases here. So let's run through all of those phases, beginning with meiosis 1. And the first phase, the phase names do not change. They're the same as mitosis, where we have prophase, metaphase, anaphase, and then telophase, followed by cytokinesis, of course. So in prophase, we start to see, here's the nucleus, this whole piece here. All of our chromosomes are inside the nucleus, and we can see the nuclear membrane right here begins to break down. At that point, we start to see individual sections of DNA become visible, and in this particular part, a phenomenon known as crossing over is going to take place, and I'll say a little bit more about that in a minute. But we can have a combination or a mixing of traits from one chromosome to an unrelated chromosome that can happen. And that's where the crossing over comes in. All right, the next phase is metaphase 1. And remember, we've got to indicate whether we're talking about 1 or 2, because there's a bit of a difference. We see our spindle fibers have formed. We have our pairs of homologous chromosomes that align along the midline, right? Metaphase, M, middle, that's how I always remember them. And the thing to remember about this is that these chromosomes, the chromosomes that align along the midline, are totally independent. So this is where our law of independent assortment will come into play. We have no control over which one of these particular chromosomes, or which are going to be one of the 4 that happen to align here along the midline. Next phase is anaphase. Anaphase A away, that's how I always remember it. So in anaphase 1, our chromosomes get pulled again by the centromere towards the poles of the cell, right? So 2 this way, 2 that way. And then in telophase 1, we start to see the cleaving of the cell. We call this little indentation here the cleavage furrow. That is where the cell is going to disconnect, right? So where we had one cell, we will now have 2 distinct cells at this point. Right? This will be cell 1. This is going to be cell 2. All right. So those get pulled. Now, keep in mind, there's still whole homologous, chromosomes are visible to us here. Right. Then our cell is going to go into the second part of meiosis, which is meiosis 2. And it, of course, is going to begin with prophase 2. So we're going to start with those 2 cells that we ended up with before. So here's cell 1, here's cell 2. In prophase, the chromosomes are very visible. The centromeres are visible. We have our centrioles at the poles here. Here's one, here's another. Our spindle fibers are attaching at the midpoint of our cell. And this is going to happen in both of the cells at the same time. Metaphase 2, again, we're looking at cells aligning along the midline. This is actually anaphase. So what happens is in each of these cells, these chromosomes will align along the midline, and then in anaphase, which you see here, the sister chromatids get pulled apart. Now, at this point, these chromatids are chromosomes. They're going to hold a complete set of genetic material inside each of those. So we can see a chromosome, 1, 4, 5, 6, 7, 8 of them. They get pulled to the poles. Telophase, remember, we start to get that cleavage occurring. It forms what we call the cleavage furrow. It's going to separate. We duplicate all of the material inside of the cell, the cytoplasm, and so on. And the nuclear membrane starts to reform, which we see right here. Nice nucleus. And we end up then with 4 cells, and they're all haploid cells, so they're containing half the number of total chromosomes of the organism. Right? This is just a nice slide here that kind of runs you through all of the parts of meiosis. Again, remember, there has to be 2 parts, so that we will end up at the end with 4 haploid cells. Remember, we start with a diploid, 1 diploid cell. All right, so during meiosis, we have our homologs that get formed early on. We have then the splitting or pulling apart of those homologs. So a lot is going on during meiosis. This particular slide, you have some time, would be good to take a look at, but it gives you a rundown of the differences as well as similarities, really, between mitosis, which is completely diploid. We're always ending up with 2 diploid cells at the end of mitosis that are identical to each other and are identical to the cell that we began with. These are our body cells, right? We want a skin cell to produce another skin cell, and we want a muscle cell to produce another muscle cell. We don't want to have differences. But meiosis is a little bit different, right? We have, a diploid cell that at the very end, we have to end up with haploid cells because we want our sex cells or our gametes to only have half the number of total chromosomes. Otherwise, if we didn't end up with our eggs having 23 and the sperm having 23, when fertilization occurs, we would have, if they were diploid, they each had 46, we'd have an organism with 92 chromosomes, which of course is not human. So 23 in the egg, 23 in the sperm, fertilization, we get the 46, which is the number that we're looking for. Now, when drawing this, remember to be sure to make sure that you're indicating, and it's very simple drawing. You don't have to be Van Gogh or anything, but just be clear to indicate the changes and the movement that's occurring to the individual chromosomes. So lastly, I just want to say a little bit about variation, because meiosis accounts for the variation. If we didn't have meiosis to help us with variation, we would all look exactly the same, right? We would be identical to one another. And that's not what we are, or even among siblings. Sometimes siblings don't look anything alike. So meiosis accounts for that variation. And one of the ways, which I mentioned briefly before, one of those ways is through what is called crossing over. So we've used nice color here, green and yellow, to indicate the crossing over. and you can see they are separate chromosomes. So in crossing over, you have to have one part of one homologous chromosome crosses over and attaches, actually exchanges really a place with another homologous, one of its homologs, but it's a different chromosome entirely. So that's kind of where we get that. The other thing that, when we look at variation is that we're looking at, the randomness, right? If we didn't have crossing over, they say that our possibilities are really finite, of what we could come up with. 2 to the 23rd. Now, that's a big number still, but it is a very finite number. So, but with crossing over, those numbers of possibilities, and as far as variation go, become infinite. We can have a large number of things going on there. Let's continue here. So again, here's fertilization, right? Sperm carrying 23 chromosomes. The egg has its 23 chromosomes, which give us a 43 chromosomes total there. All right, just another look there. Non-disjunction, when we get right here, when we get a part of a chromosome that doesn't separate, right? That's what non-disjunction is. So we'll end up with, in the end, one of our, let's say, eggs that has too many parts. So this is n plus 1. And then we would have one that does not have enough, n minus 1. So one of the non-disjunctions is, when we see trisomy 21, there are 3 chromosomes on that 21st pair with Down syndrome. So that's what that happens to be here, non-disjunction. As long as we're talking Down syndrome, usually around age 40, look right here, a mother who's having a child, especially the first child, at age 40 or other, you can see, or older, you can see how exponentially the odds of having non-disjunction occur in that female